Hello, welcome fellow IGEMers and people interested in science. I am the team manager leader of Links China, an IGEM team from the high school division. The subword link from our team name is to represent a versatile background for our talented members. We have coding and designing enthusiasts from Shenzhen local and international schools. We have article referencing and model constructing geeks from American boarding schools, and most importantly, a large united laboratory group from both backgrounds. Although the pandemic made it hard for all of us to gather in one physical location to work, we want to make the optimal outcome through the internet.
For this year's IGEM project, our team in Shenzhen, China decided to use synthetic biology to solve a problem within our local community. One of the biggest problems we came up across was the problem of electrical energy consumption. Because up until about 2017, the primary way to get electrical energy was by using nuclear or natural gas. This poses a great risk to our environment, and it's not sustainable in the long run. So our team thought that if we use synthetic biology to create a more eco-friendly alternative solution, it could be a great way to influence our environment in the good ways. Good thing is, we came across a Nature article which detailed how a group of scientists were able to create voltage from air. They did this by using a nanowire film that was made of pillins from Geobacter theoreducens. These pillins are able to produce a current from humidity in the air and ultimately use it like a battery. So we thought, if we were able to replicate the same system within E. coli, which is a much more lab-friendly and studied bacteria, then we would be able to optimize the different factors within the system and ultimately produce a product. So our goal for this year is to make a biobattery that would be able to work in different environments such as rainforests or deserts, and maybe even work in daily life to replace conventional batteries like button batteries. With no toxic waste and a green energy footprint, we could ultimately provide a great alternative to our current energy crisis. So let's see what's going on in the lab.
First, we will dilute the supernatant containing bacteria and pili. Next, we transfer the diluted supernatant into the filter tubes. Insert the supernatant into the centrifuge. Place the test tubes symmetrically to maintain balance. After centrifugation, take out the filter tubes and observe whether there is visible pili precipitation at the bottom. Using a pipette to transfer the liquid left in the filter into the EP tube. Divide the liquid and add protein loading. Put the solution into the steamer. It will separate the tangled protein into a single chain structure. Now we will perform electrophoresis. We take out the gel that we have made and begin assemble the necessary parts together to make a container. Now we'll pour in the TAE solution. We will push the solution slowly into the gel through the pipette. Now we will begin electrophoresis. Then we will prepare to transfer. using a tweezer to press the membrane and glue on 3 layers of filter papers to make them stick together more tightly, ensuring the protein could be transferred into the membrane. Put the whole block into the transfer tank and be sure the colors are matching. Pour into the transfer liquid and add ice cubes because the high current in the system will release heat that would potentially denature these fragile proteins, and the bindings between the membrane and protein may also be affected. And now we will turn on the electrofield again. Now we will retrieve the membrane, put it into the blocking box, and leave the box in the shaker overnight. The fluid is aspirated with pipette. And a primary antibody is added to make it adhere to the protein with tag. The next day, we will incubate the box for 1 hour and then aspirate the solution out with a pipette. The incubation of a secondary antibody binds to the primary antibody, and since the secondary antibody has a staining group, it stains the protein with tag. Now we will add the dye that could help us detect our target protein. And this is a whole process of our experiment of identifying GS-pili protein sequence. And future experiments will be continued.
我们现在已经做到了体外表达的这些项目，就是我们前面已经构建好了智力，然后现在开始体系菌毛。我们希望尽快可以出现结果，这样子实验就已经算一个非常大的成功。然后希望大家也可以多多关注我们接下来的实验。在实验室就是能感受到很多新鲜的东西，类似于以前从来没有做过这种生物实验，但是现在可以感受一下。然后呢，虽然实验步骤有很多就是比较难，但是也比较累的东西，但是从头到尾都是还是比较开心的。
我最 fascinated 的一点对于 IGEM 就是我可以改造微生物，然后让他们去产我们所要的一些 product， 然后从而我们就可以对我们的生活啊，或者说对科技都会带来改变。对我们的项目是非常充满信心的，而且我觉得我们项目是应用前景非常广，然后它也是非常具有潜力的一个项目。最后呢，我希望大家可以对我们的项目有多多关注，然后我也非常欢迎其他队伍跟我们一起来 collaboration。Let's make the future better.